to be on several items. I'm going to start off by talking about good clinical practice, and from good clinical practice based upon the principles of International Conference on Harmonization. And then I'm going to talk about the definition of substantial evidence of effectiveness. When we want a drug to be effective, the evidence must be substantial, so we need to define what that means. And then I'm going to talk about elements of clinical trial design, the purpose of clinical trials, the types of clinical trials, the patient populations, the primary efficacy endpoint definition, as well as secondary efficacy endpoint definitions. I'm going to talk about statistical versus clinical significance and the difference between the two, and what it means when one says we have to have adequate alpha distribution. Then I'll talk about data elements for a substantive review, some review metrics, subgroup analyses, and ultimately the benefit risk analysis, which is the determinant factor in how a drug is approved. So for good clinical practice, focusing on the principles of International Conference on Harmonization, there's a long history to this that goes back to 1943, where studies were done that did not consider the benefit of the patient, and guidelines were developed with consideration of the current good clinical practice paradigm, and the United States, Canada, the European Union, and non-European Nordic countries, Japan, Australia, and the World Health Organization all collaborated to produce a set of rules on how to maintain good clinical practice. The ICH, or International Committee on Harmonization, passed this good clinical practice called E6 in 1996. It was adopted as an FDA guidance in 1997, and it applies to clinical trials that are intended to be submitted to regulatory authorities. Now, clinical trials should be conducted in accordance with the ethical principles that have had their origin in the Declaration of Helsinki and that are consistent with good clinical practice and the applicable regulatory requirements. Before a trial is started, the risks of the trial and the inconveniences to the patient need to be weighed against the potential benefit for the individual subjects in the trial and for society as a whole. A trial should be started and continued only if the anticipated benefits justify the risks. The rights, the safety, and the well-being of the trial subjects are most important considerations and should prevail over interests of science and society. And clinical trials should be scientifically sound and described in a clear, detailed protocol. A trial should comply with the protocol that has received prior institutional review board and independent ethics committee approval. And individuals involved in conducting a study should be qualified by education, training, and experience to perform everything we know about the drug, all the risks, all the potential benefits. All clinical trial information should be recorded, handled, and stored in a way that allows its accurate reporting, interpretation, and verification. The confidentiality of records that could identify subjects should be protected, respecting the privacy and confidentiality rules in accordance with applicable regulatory requirements. The investigational product should be manufactured, handled, and stored in accordance with applicable good manufacturing practice. They should be used in accordance with approved protocols. And the systems with procedures that assure the quality of every aspect of the trial should be implemented. So in summary, the key messages for good clinical practice based on ICH E6 is that every trial is designed to ensure the right, safety, and well-being of trial subjects and making sure that they are protected. A trial should be initiated and continued only if the anticipated benefits justify the risks. The data and reported trial results should be credible and accurate. Clinical trials should be scientifically sound and described in a clear, detailed protocol. So this leads to obtaining substantial evidence of effectiveness generated from the clinical trials. So now I'm going to go into what exactly is substantial evidence of effectiveness. I'm going to go back to 1962, where the Food, Drug, and Cosmetic Act was formulated in law. And I'm going to read this legal statute to you. It is a substantial evidence definition as evidence consisting of an adequate and well-controlled investigations, and that's plural, not one but two trials, that's what it's interpreted to be, including clinical investigations by experts qualified by scientific training and experience to evaluate the effectiveness of the drug involved, on the basis of which it could be fairly and responsibly be concluded by such experts that the drug will have the effect it purports or is represented to have under the conditions of use prescribed, recommended, suggested in the labeling or proposed labeling thereof. So in other words, in order to claim that a drug is safe and effective, you have to do good studies to prove it, and it has to be done by people who know what they're doing. And that's the law. And then the law was translated into practical application. The Code of Federal Regulations, 20 CFR, states that the purpose of an adequate and well-controlled investigation is to distinguish the effect of drug from other influences, such as spontaneous changes in the course of the disease, the placebo effect, or biased observation. So when you do a clinical trial, the trial has to be designed in such a way that the difference between a drug and placebo is due to the drug, not to any other factors that might have interfered with the outcome of the study. The characteristics of an adequate and well-controlled lack of effect in a control group or compelling mechanistic evidence in the setting of a well-understood disease pathophysiology or a well-documented natural history of the disease, and scientific knowledge. That one trial needs to be supplemented by confirmatory evidence. Otherwise, you do two trials. Now, confirmatory evidence could be waived if the result of a single trial is so good, so persuasive, so clinically meaningful, like you decrease mortality or you decrease irreversible morbidity, you can waive confirmatory evidence. Now, confirmatory evidence is required if the results are positive but not overwhelming, not based on mortality or irreversible morbidity, such as feel, function, and survive, patient-related outcome, like telling a patient telling you, I feel good today, or I can exercise better. Those are not the same as mortality and or irreversible morbidity. So the point here, the key messages of effective evidence of substantial evidence of effectiveness is that substantial evidence of effectiveness is defined as a demonstration from clinical trials that a drug conclusively and unambiguously produces the treatment effect hypothesized in the clinical trial. The results are not confounded by other variables that may have impacted the observed treatment effect. The observed treatment effect must be clinically meaningful and statistically significant. That is a low chance that the results are a false positive or a false negative. To achieve this, the clinical trial should be properly designed. So now this leads to learning about how to design clinical trials. What is the purpose of a clinical trial? It's obvious. The purpose of a clinical trial is to determine if a new medical for the trial to be considered positive. Now, what do all good clinical trials have in common? They are all adequately designed. The trial should discern the purported treatment effect. They should be well controlled, usually a randomized trial that attenuates based on imbalances of patient characteristics. If you have 100 patients and they get equally divided randomly between drug versus control, chances are all of the characteristics about this patient population will be equally divided in a well-controlled and randomized fashion. The outcome of the trial needs to be interpretable. Data from the trial must be of high quality for a definitive conclusion to be drawn with respect to the hypothesis. And there needs to be a pre-specified criteria for success. A trial design should include pre-specified acceptability of a type 1 false positive or type 2 false negative error. So nothing is ever perfect, but we always assume that there's a small chance you're going to have a false positive. You think the drug works when it really doesn't, or a false negative, that you prove that the drug doesn't work when in fact it does work. You have to accept a small chance of that, and that criteria is the basis of a statistical analysis plan. So again, all good trials are adequately designed, well controlled, the data is interpretable, and there's always a pre-specified criteria for success. Pre-specified means that before you even start the trial, you are setting the rules, as opposed to setting the rules later on, when in the middle of the trial or at the end of the trial, where there's a very good chance of bias that might have equally prescribed. It is useful in long-term extension trials to evaluate safety or persistence of effect. The cons of this open label strategy is that there's a possibility of bias. Patients or the investigator may have a preconceived notion of safety and or efficacy that might impact this. The con of a blinded trial is that it's very costly. More complicated logistical designs in terms of randomized code generation, blinded study drug kit design, distribution of study drug kits, treatment assignments, monitoring for study drug allocation assignment. It's a much more costly and complicated trial to maintain the blindness. But most importantly, the expense is worth it because the results will be interpretable. Now, there are different types of blinding strategies. There's a single blind, the double blind, and the double blind, double dummy. A single blind is when the subject does not know what he or she is being given, but the investigator knows. Double blind means neither the investigator nor the subject knows what is being administered. Now, double
So you're on mute. Can you unmute yourself? You're on mute, sir.
Oh, can you hear me now?
Yeah. Yeah. You're audible. Thank you so much.
Oh, have I been not heard the entire time?
No, just for a few seconds, sir.
Oh, okay. Thank you. Only, and one is placebo. So, and the reason why one would do this sort of a trial is to test whether or not a drug combination is superior to individual drug components. Now you have the crossover trial. This is a trial where patients are randomized to treatment A versus treatment B, then similar size parallel group trials. This allows you to get good pharmacokinetic data and to make sure that all of the clinical data like pharmacokinetics, pharmacodynamics are understood prior to a phase 2 or phase 3 trial. Now we have the placebo-controlled withdrawal trial. This is a situation where everybody takes a drug and then they get randomized to either maintaining the drug or to be given placebo That means they're taken off the drug. And if once they're taken off the drug, then whatever disease they had that got them into the trial, the symptoms will return. And when you have a return of symptoms when patients were randomized to placebo, and maintaining lack of symptomatology when patients were maintained on the drug, that difference represents the treatment effect. So the primary efficacy endpoint is a relapse of symptoms following withdrawal of a drug. The trial design has been used for most likely hypertension studies and studies for pericarditis, chest pain, because the membrane that surrounds the heart has been inflamed. Now, the patient population is very important. There are three types of populations that you will hear when you talk about clinical trials. One is called the intention to treat population. That is, everybody who's been randomized to a trial, regardless of whether or not they've been given the drug, is called the intention to treat population. This represents the real world. Then there's something called the modified intention to treat population. That is, the randomized subjects who took at least one dose of study drug. That's what's called modified. And then there's the per protocol population. This is a population of patients who followed the protocol exactly as written without any protocol deviations. Now, the ITT, the intention to treat population, is preferred. The intention to treat analysis preserves the prognostic balance afforded by randomization, thereby reducing any risk of bias that may be introduced by comparing groups that differ in prognostic variables. So if you start not analyzing the intention to treat patients who dropped out versus patients who did not drop out, there could be a difference. And you could affect the interpretation of your data if you only look at patients who were in the study and not those who dropped out. The modified intention to treat population is okay, it's tolerable. The risks here are excluding subjects that could introduce bias and lead to misleading results. And the per protocol population is seldom endorsed. So if you're working for a drug company and you want to propose, want to propose a per protocol population as the population you will analyze, by excluding subjects who deviated from the protocol, it introduces attrition bias in which groups of subjects being compared no longer have similar characteristics. Now, who do you include in the clinical trial? You include subjects that are likely to benefit from the drug, from the intervention. You don't want them to be too sick, because if the subjects are too sick, it's maybe too late to treat the disease. You don't want them to be too well, on the other hand. If you're so well, the drug is not going to have much of a treatment effect. So you want the patient population to be just about right. And you could have, unfortunately, something called a regression to the mean. And what that means, a regression to the mean, you could select a subject experiencing a bad day or having a good day at baseline. So when a subject comes to you and they sign consent and they're in a clinical trial, they could be having a bad day, or they could be having a good day, they feel well. Such a subject may revert to their average level of illness during the trial. So if they're having a really bad day when they got randomized, and then a couple of days later they feel better, it's not because the drug did anything, but it might be because they are back to what they used to be before they had a bad day. Or if the subject is having a great day, and he very seldom has a great day, but he happens to have one of those good days when he signs consent and gets randomized, and then a few days later he goes back to the way he was, you're disease presentation. And the best defense of this is a control group in a randomized trial. So always go back. The clinical trial is designed to test the hypothesis that the drug reduces the incidence of a primary endpoint by a pre-specified amount. The statistical analysis plan, euphemistically known as SAP, statistical analysis plan, including sample size, focuses on being able to draw conclusions regarding primary endpoint. The primary endpoint could be a single variable, like mortality, or it could be a composite of several variables, like cardiovascular death, myocardial infarction, or stroke. Now, one important thing to note here is that when you have a composite endpoint of several variables, each variable should have the same weight as the other variables, like death, heart attack, and stroke. They're all serious. And not to sound funny, but um, if you had death, stroke, and hair loss as a third variable, where the clinical study will be driven by how much hair you lost over the past 5 years, and it doesn't say anything about death and stroke, that is a bad endpoint. And I use that as an extreme example of what constitutes a good composite endpoint. Now, secondary endpoints are usually specified as a matter of scientific interest. The trial design focuses on the primary efficacy endpoint, but sometimes a drug company likes to have their secondary efficacy endpoints as part of their label. They want to be able to make claims. So if that is the case, if key secondary endpoints are planned to be included in the drug label, then their testing should be included in a statistical hierarchical strategy to attenuate the chance of producing a false positive result. So let me give you some key messages on trial design. All trials should be adequately designed to answer the primary hypothesis, should be well controlled, have interpretable results, and have a pre-specified criteria for success via an adequate statistical analysis plan. You have to pick the right protocol design to fit the program hypothesis. The gold standard is randomized double-blind clinical trials. It attenuates influences of confounding variates, and it minimizes the potential for bias. If secondary endpoints are planned to be included into the label, the statistical analysis plan should include an hierarchical strategy designed to avoid creating a possibility of producing a false positive result. Now, this leads to a discussion of statistics. So I'm going to turn to statistics. What is statistical versus clinical significance? And what is the difference between each assessment? Let me start off by saying that statistics is a tool that sets boundaries for determining efficacy. If you cross a line, you're no longer effective. Alpha, we use the word alpha to define the probability of committing a type 1 error. The standard alpha is 0.05. The whole world accepts 0.05 as alpha. That is to say, a 5% chance of committing a type 1 error. That is to say, a 5% chance that you rejected the null hypothesis when it was true, false positive. When you have a hypothesis, my drug works, or better than placebo, the null hypothesis is my drug does not work compared to placebo. When you reject that null hypothesis when it's true, it's a false positive. The drug doesn't really work and you think it does. So there's a belief in the results with 95% confidence. Beta is the probability of committing a type 2 error. Now, we usually set beta as 0.2, 20%. And the reason why we set beta as 20% because we want to set alpha at 5%. Now, let me give you an analogy. In a court of law, when you're on a jury and a person is on trial, you want to make very sure that the person does not get found guilty when they're actually innocent. That's a false positive. That is a type 1 error. So if you want to make extremely sure that you will never convict an innocent person, you stand a chance of not convicting some guilty people. They go together. So when you set alpha of 0.05, setting beta at 0.2 represents a good balance so that you have a clinical trial with a reasonable sample size. If I want to be 99% sure, not 95% sure, setting alpha as 0.01 or 0.005, then that beta is going to be very, very high. So you're standing the chance of proving that a drug really doesn't work when it actually does. So you have to set the balance to everyone's satisfaction. So alpha is set at 0.05 and beta is set at 0.2. 20% chance of committing a type 2 error. 1 minus beta is the power of a trial. So when you say you're going to power the study and you want 80% power to see a difference, 80% power is 1 minus beta. That is, you're accepting a 20. You're going to get a statistically significant difference of 1 millimeter of mercury, but it has no clinical relevance whatsoever. So there has to be a combination of statistical significance and clinical relevance. So clinical significance is the magnitude of a treatment effect that would convince many doctors to alter their practice. This is different from statistical significance, and it incorporates a broader issue of benefit risk. An example here is a drug shows a persuasively clinically meaningful effect in a rare disease with an unmet medical need, clinically meaningful effect in a rare disease with an unmet medical need within the pre-specified criteria for success. That pre-specified criteria points being included in the label. We set alpha as 0.05. That is the standard. Now, let us say a drug company comes to you and they say, my primary endpoint is exercise capacity, and my secondary endpoints are how a patient feels and how many times they went to the hospital. And they want all three of them in the label. So in order to make sure that you don't make a mistake and commit a type 1 error if you test so many times, you set the alpha at 0.05 and you distribute it amongst the variables that are designed to be in the label. So you might say, out of the 0.05, half of that, 0.025, will be the primary endpoint. And then you're going to take 1/2 of the other 0.025, and they should be 0.0125 rather than 0.025. That's a typographical error in my slide, and I apologize for that. But all of the alphas should add up to 0.05. Now, in order to make sure that you have the data that you need to conduct an analysis that allows you to draw the conclusion, there are 4 elements of integrity issues. So again, the 4 elements for a substantive review: how the data was collected, how it was analyzed, is the data stable, and is there data integrity? And there are things that you're going to look at when you look at it, the results of a clinical trial, to determine whether or not there's substantial evidence of effectiveness. How long was a trial? Was it long enough to see the effect that is based on the proposed claim? Is it the right patient population? Was the primary efficacy endpoint agreed upon? If the results are statistically significant, is it clinically meaningful? What was the randomization scheme? It's supposed to be completely plan, a data safety monitoring board, plan, a data safety monitoring board, an adjudication charter, an adjudication plan. Was the governance of the trial adequate to do the job? So these are the things that you look at to ascertain whether or not a trial was well done and the results are substantial. Now, I'm going to heuristics: age, gender, race, and ethnicity, what region in the world, whether or not they smoked or had diabetes, or had a past history of myocardial infarction, or had a history of cardiac revascularization. The key question here about subgroup analyses: are the results the same for each subgroup? Are the results of the trial driven by only one subgroup? Is there a subgroup for which the drug is harmful? So a subgroup analysis facilitates the identification of what drove the efficacy results and whether there is a safety concern for a specific subgroup. Now, a couple of notes here. Efficacy interpretation. A positive trial driven by only one region of the world, or one country, or one site is still a positive study. This happens quite a bit. The study could be negative in the United States. It could be negative in Western Europe. It could be neutral in India, and it's highly positive in the Ukraine. And if you took out the Ukraine, the study was negative. Well, that's data fragility, but it's still a positive study. A negative trial with a strongly positive result in one region is still negative. So if the trial was highly positive, was negative, but one section of the world showed highly positive results, it is still a negative study. Now, too many subgroups can cause a type 1 error. It's like you want to throw a dice and you want to hit a 7. And if you throw the dice twice, the chances of hitting 7 are not so high. But if you throw the dice 100 times, there's a really good chance of hitting 7 at least one of those times. So there's a study that was done that shows the more subgroups you look at, the greater the probability of having a false positive study. That's what this graph shows you. But despite all the misgivings of a subgroup analysis, it does have its benefits. I'm going to focus on that little red circle. This was a study about a drug that makes your platelets not able to clot. So this is an anti-clot drug, and the name of the trial was called Plato for platelet aggregation studies. Now you might call ticagrelor. On the other hand, if you take a baby aspirin, 83 milligrams, then the drug works with the lower dose of aspirin. So subgroup analyses does have its place because it identify a risk, and that could you can put a risk warning in the label. So it's good to do subgroup analyses, but be careful when one does it. Now the ticagrelor, an antiplatelet drug. Since I'm a cardiologist, I go for these types of drugs. And the efficacy of this drug is cardiovascular death, type 1 myocardial infarction. That is, you get a big heart attack in the street after you've been taking this drug for some time. Oh, I'm sorry, these are the benefits. You're reducing death, you're reducing heart attack, and you're reducing stroke. But since you're inhibiting platelets, you're going to get a bleed. A bleed could be fatal. A bleed could be a brain bleed. You're getting a stroke. Or you could lose so much blood that you need a big transfusion. So bleeds are the risk. And those three boxes at the bottom, the fatal bleed, the intracranial hemorrhage, and the too much bleeding, I'm going to call them TIMI major bleed. That is the title of those three bleeds combined. And then the benefit is I'm reducing death, heart attack, and stroke. And I reduced death, heart attack, and stroke by 18%. The hazard ratio was 80.82, and it was statistically significant. The risk showed excessive bleeds, but luckily none of the bleeds were fatal. Now, this is a little bit of a calculation. How many patients do you need to treat in order to get one benefit? And one benefit is defined as one of those components of the composite endpoint. And the number of needed to treat, now in the ticagrelor arm, the event rate for the primary efficacy endpoint was 6.9%, and in the placebo arm, it was 7.6%. That was statistically significant. So if you divide the placebo rate by and you divide the 1 divided by the difference between the placebo rate and the ticagrelor rate, you get 143. That is to say, you need to treat 143 patients to get one benefit. That is a reduction in one of those components of the composite endpoint. How many patients do you need to treat to get a harm, a major bleed? Well, the placebo arm had 100 bleeds. The ticagrelor had twice as many. And so the number of needed to do one harm is 67. So actually, you need more patients for a benefit than you need for a harm. In fact, for every patient that has a benefit, there are 2 that are having a bleed. And that is the basis of your risk-benefit evaluation. There's some trouble with that. When the number of needed to treat um, is greater than the number of needed to harm, that is to say, more patients will be harmed than be benefited by, you run into trouble. But how important is a death, heart attack, and stroke relative to a bleed? Now, if the bleed kills you, it's just as bad as if you died from a heart attack or stroke. If the bleed doesn't kill you and you get a transfusion, you could survive it. So there is more than simple number of needed to treat. You need a weight that weighs the importance of each of these variables that go into the benefit versus the risk. And on top of that, this is a new thing that's been going on with the FDA. It's called the multi-criteria decision analysis. And in plain English, that means we are looking to see how many people feel about the benefit and the risk. What does the patient feel? Some patients would say, I would rather be dead than have a stroke. I don't want to bleed. I don't want to go to a hospital bleeding all the time. I'd rather just have my heart attack. Some people might say that. So you need to take into consideration patient preferences when establishing a bridge algorithm for a benefit risk evaluation. So I'm going to conclude this talk by some key take-home messages. Number one, clinical trials are designed under good clinical practice principles as outlined in the ICH E6 guidance. Number two, recommendations about approving a drug rest on availability of substantial evidence of effectiveness that is statistically significant and clinically meaningful. Number three, obtaining substantial evidence requires an adequate trial design, identifying the correct patient population, and identifying a primary endpoint that reflects the purported treatment effect detectable by the patients. Number four, primary and secondary endpoints planned to be included in the label should be part of an alpha conserving strategy to avoid increasing the chance of committing a type 1 error. Number five, assessing data requires knowledge of how it was collected, how it was analyzed, and how stable and fragile the data is, and if there is a loss of data integrity because of missing data. And finally, the benefit of a drug needs to be weighed against the risk of the drug, considering the weight of the benefit and the weight of risk, as well as patient preferences. And that concludes my presentation. And thank you for your patience in listening to this nonstop talk that I just gave.
W- Thank you so much, sir. That was really impressive. And
I'll be happy to entertain any questions one might have.
thank you so much, sir. That was really informative. We have our principal here, Dr. Ramesh Alluri. He just want to speak a few words, if you allow him.
Absolutely. Thank you.
Uh, Thank you so much, sir, for your wonderful presentation. And yeah, uh, I think I was really so happy to learn from you. Thank you. I think you are a person with multidisciplinary. When I've gone through your bio, right, you sailed from a chemical engineering to, and you mastered in the sciences and shown your presence in the clinical trials. And I think really very few people of your will have this caliber to convert themselves into this way. I think thank you so much for spending your valuable time. I know it's very late for you, and very sorry for making you late despite your little health condition. I think thank you all. I think it's almost 11 o'clock at night, I believe, sir, for you.
Midnight,
11:30
11,
midnight. Okay.
yeah. But
I think-
I want to thank you for the
Yeah.
privilege of having spoken to you, and I hope to do it again one day.
I think first we should thank our Jagdish sir for connecting us, and I have been sharing. I will be happy to meet you in person there whenever I come to US, and I think definitely we'll take this forward for the next coming series sessions, and we want you to be there and bless our students with all your knowledge. And thank you so much for your presence. And I
Uh,
think it's over to the questions. Thank you. Please, sir.
The room is open for questions. Good evening, sir. This is Lavanya from final year. I have a question. How can we be sure that the results of the study are reliable and reproducible? Is that entirely going to depend on risk benefit analysis, or are there any parameters?
Now, reliability and reproducibility is not based on benefit risk, but it's based on sensitivity analyses. So when a drug company gives you the data, they give you a data set, and you need to be able, this is what we require in the United States. In Europe, it's a little different, but we have to be able to reproduce the data. So we look at the patient, we look at the statistical analysis plan, we take the raw data, the actual data gotten from the patient, and we analyze the data ourselves using the same plan that they said they used and see if we get the same results. And if we do, then it's reliable. If we do sensitivity analyses, let's take a few hospitals out of circulation, and let's see if the data maintains its stability or falls apart. And if we start noticing that the whole result was driven by one, even though the company may not have said that, but we uncovered it by our own sensitivity analyses, then we would have some idea of how unstable these data are. So reliability is based upon our own independent analysis and our own sensitivity analyses.
Thank you so much.
Yeah, I have a question, doctor. Uh, Can I know the way you have proceeded your clinical trials during the corona, and
Hmm.
how did you come up with the vaccine at a very accelerated state?
Sorry for my cough. There was a couple of things that happened. The Department of Health, the Human Health Services, HHS. This is the division in the executive government that the FDA reports into. The secretary declared an emergency. So this was not like a expedited review, but this was called an emergency, which means that we are mandated to cut our review time. So when a company comes and wants to do a study on a drug that is purported to reduce the symptoms of COVID, instead of reviewing it for 30 days, we review it for 5 days. And we are more flexible on the benefit risk because people were dying. And so we said yes more often than not. When it came to the vaccines were also part of the emergency use authorization. And all we knew was that the vaccine was producing uh, antiviral antibodies that were neutralizing. It was actually killing the virus, but we did not know how long it would be effective. We did not know if there were any side effects. So this was basically one huge experimentation. And when someone took the vaccine during the coronavirus, the informed consent had to say, we don't know as much about this vaccine as we would normally know when a vaccine is approved. So everything was expedited, and it was also conducted where the full knowledge of the characteristics of the vaccine were not there. And that's how we did it. And also for clinical trials that were going on at this time, the coronavirus pandemic erupted, we had to change the way we look at data. Patients coming to a clinic was no longer a feasible thing. And we had to now devise ways to measure variables like exercise capacity, heart rate, blood pressure from remote um, remotely in order to get the data we need to ascertain the efficacy of a drug under those conditions where coming to the clinic was harmful. And that's how we were functioning back then.
Thank you, sir.
Asking one question, sir. Yeah. Sir, good evening once again, sir. I was just about to how we need to overcome the challenge of rare adverse effects which will be doesn't happen at clinical trials. For example, if you take a flu vaccine, pandemics, 1 in 12,000 of patients they are affected with narcolepsy. So how could be the significance of rule of three come and how to overcome these challenges when to overcome, as you said, the clinical trial, the substantial evidence what the clinical trials will provide, how to overcome these type of challenges and
Yeah,
prove.
that's a difficult
Yeah,
thing. When you have a rare adverse event, you need a huge sample size to detect it.
exactly.
And
Exactly.
one example that I dealt with when we were getting reports of myocarditis, and the incidence was low, but I went to the National Institute of Health and I looked at the database, and it turns out that people normally get myocarditis, low incidence, and every age bracket, up to 0 to 10, 10 to 15, 15 to 20, 20 to 25 years of age, and so on. There's a certain rate that if someone gets myocarditis is within what we already know epidemiologically. It turns out that in a particular age group from 18 to 25 years of age, there was a slightly higher incidence of myocarditis than the 95% confidence interval dictates. And we didn't know whether or not the vaccine was doing it. So we called Pfizer and we said, I think your vaccine is causing myocarditis. They did a study where they looked at every protein on the surface of the coronavirus, and they looked at every protein on the surface of the heart to see if these proteins look like each other so that the antibodies that are being produced by the vaccine are going to attack the heart. And they found no overlap. So one way to overcome it is to do some studies molecularly to see if there is a pathophysiological pathway to ascertain whether or not there's a feasibility of some mechanistic induced or iatrogenically induced adverse event. But if you have 1 in 12,000, you're not going to see adverse events that rare unless you have a lot of people taking the drug or the vaccine. And that's a difficult situation. It kind of comes up on you after many years.
Yeah. Thank you for that. And one last question. As you're an expertise, of course, it's an irrelevant for you. Present talk might be as an expert of artificial organ transplant. So, what could be the future of something quite interesting? So, we would like to just throw a light on that particular area for a
Mm.
minute. Thanks.
When I was very young and I was an engineer, I was so passionate about artificial organs, artificial heart. And it turns out that anytime you put anything artificial in the human body, the body rejects it. Blood will clot around it. And we began to look at ways to stop blood from clotting in an artificial organ. And so I began to coat the inside surface with polymers that would hold heparin, for example. And then the science changed from something artificial and mechanical to growing a new heart, growing a new organ using stem cells that have been re-engineered to form a new organ. And that's where we're stuck at this point. One particular initiative was the vascular endothelial growth factor that would produce new blood vessels to bypass the clogged arteries in coronary artery disease. And so organ regeneration and blood vessel revascularization are the trends that we see occurring right now.
Thank you very much, Dr. Fred. It was so nice to hear from you this morning here. And Fred and myself, we published two papers on angiotensin 2 septic shock. In 2019, I think angiotensin was approved for septic shock. Then also we were together on vasopressin for septic shock, followed by review article on septic shock and pharmacological research. And then Dr. Fred also associated with me in editing this mega book, The Quintessence. Recall that we spent about nearly 1 year in collecting, writing to people, and soliciting chapters. Although it was planned for 62, finally three dropped out. We had 59 chapters, a mega book, and that was published last year, that is October 2023. It was so nice. Thank you so much, Dr. Fred. It is already nearing midnight for you to be there. Thank you. I appreciate your time.
Thank you so much, and have a wonderful rest of the day. And until next time, thank you.
Sir, hello.
Hi.
Sir, I have one more question. One more
Mm.
question. That is, what are inclusion and exclusion criteria in clinical trials?
Oh, inclusion and exclusion criteria in clinical trials are what does the patient need to have in order to make it into the study? That's the inclusion criteria. They have to have the disease. They can't be too sick, or else they're not going to the drug won't be effective. They can't be too well. So there are a bunch of criteria that allows the patient to get into the trial. Exclusion criteria are those that could affect the patient, make it not safe for the patient to be in the trial, or it would confound the results. For example, if you're testing somebody for high blood pressure, you're developing a new blood pressure drug, and then you find out they have terminal cancer and they'll be dead within 3 months. That's an exclusion criteria. They can't be in the study. They won't last in the study. So inclusion is what are those aspects of the patient characteristics that make them eligible for the trial? Exclusion, what are those things that would make them ineligible for the trial?
Thank you.
Thank you, Dr. Fred Senator. I would like to express my sincere gratitude for your enlightening presentation on clinical trials. Your expertise and clarity in explaining the complex concepts of medical research were truly impressive. We appreciate your commitment to share your knowledge even while feeling unwell and the odd hours. Thank you once again for your valuable contribution to Vishnu Institute of Pharmaceutical Education and Research. Thank you,
My
sir.
my pleasure. Thank you so much. Okay. Have a good rest of the day, and until next time. Thank you.
Thank you so much. One.
Message to him that
Yes. Yes.
so is it for 10:45? So we can take coffee and we will go for breakfast.
Please take break for 15 minutes.
Finally.
अच्छा ready होंगे sir? Yes, sir.
Yeah. Okay.
Oh, Nabakaman is
Yeah.
ready. If you're ready,
He's ready.
we can start it.
Yeah. I think you can start.
Other people get up and go. If anybody-
Yes.
वालेकुम अस्सलाम
Please be silent for
Please maintain the decorum for 5 minutes. The session is going to start.
Good morning, Dr. Balakumar Piche, sir.
Good morning.
Yes, sir, you can start the session now.
Yeah. Am I audible?
Yes, sir.
Okay, thank you very much. So good morning, everyone. Good morning, organizer, Professor Ramesh, Professor Rajasekhar, and everyone. Nice meeting you again. So the title of my presentation for today is Mastering Manuscript Submission and Strategies to Avoid Desk Rejection. So this is what I'm going to discuss for another 40-50 minutes. So most of the contents that I'm going to present that are coming from the book yesterday, as we told, the quintessence of basic and clinical research and scientific publishing. So as I said yesterday, scientists must not only do science, but must also write science. So we need to develop our skills, writing a paper for its successful publication in Web of Science, Science Citation Index Expanded, Q1 top tier journals. So such kind of skills we need to develop, that is very much inevitable. So this is the slide that shows publish or perish, a call to action. So we start the research with idea, and many people they leave the research with idea only. So it means they will be perished. No more will be in the academic. Some people they take the idea into research and they do the original research, but they are unable to write the research. It means they will be perished. No more will be in the academic. Some people, they write the research, but unable to publish it. If they are unable to publish it, again, they will be perished. They will no more be in the academic. And a very few people, they come up with the idea, they do a good research in academic at the PG level, they write the paper and they publish it. Once you publish your paper, it does not mean that your task is done. As long as you are in academic, you have to get it repeated again and again. You have to come up with a new idea, a new research, a new paper, a new journal publication. Again, you have to repeat it. So as per the NAC, it says that every faculty member should publish 2 papers per year and 10 papers for 5 years. So then only we can get a full mark, 80 marks for number of publications and 80 marks for citation and 80 marks for H index. When we reach H index 35, we will be able to get full mark. So the publish or perish action is very much, it needs attention in academic. Once upon a time, a teacher means has to only teach. But nowadays, a teacher means not only to teach, so do also a original research. Otherwise, a teacher cannot become a teacher now. So this is very much important for every teacher, even right from assistant professor to guide the students, to do the original research, to come up with ideas and convert ideas into research, write the paper and get it published in a journal of international repute. That is again and again I'm telling, Web of Science, Science Citation Index Expanded, Q1 or Q2 or Q3 or Q4, or at least Scopus Index Journal or PubMed Medline. Automatically, most of the PubMed Medline Index journals are coming under Web of Science. So there will be always a pressure from the higher authority, like Professor Ramesh is sitting. So he has to do this task. Otherwise, we cannot get a target. We cannot achieve the target. So such kind of action is very much important. We need to give some kind of pressure, some kind of force. So then only the people are moving forward to do the research. Otherwise, it is very difficult to achieve the target. So yesterday I told you more than 10,000 papers were retracted in last year itself. It is because of scientific misconduct. You should never cite in your paper any retracted paper. For example, you are citing 40 references in your paper. Make sure that 40 out of 40 are free from any retracted sources. even if it is 4 or 5 papers are from retracted sources that you have referred, and your paper will likely be desk rejected. So you have to be very careful. For example, you are writing an original research article and you are citing 50 references in the list. Out of 50, for example, 7 references are retracted that you have referred. It means you have also done some mistake, and your interpretation also may end up with some mistake. So you should make sure that not even a single paper that you have listed in your references are retracted. So it is a word of caution. It's a threat to scientific integrity, and also make sure that you try to avoid citing any paper mill paper. Paper mill you can easily identify as far as your experiences goes. So you should never try to cite any kind of papers coming from paper mill, because that paper mill, when your paper is being processed, may be retracted, that paper. So to avoid such kind of inconveniences, you should try to avoid any papers to be cited through paper mill. So coming to the point, at the end of this presentation, the participants will be able to explain the ways to avoid desk rejection. I do not want you to understand. I want you to explain at the end of the session. So it is a higher order cognition. And also the participants shall be able to explain the tips on successfully publishing a manuscript in Web of Science, Science Citation Index Expanded, Q1 or Q2 or Q3 or Q4 journals. So before you start writing any paper, make sure that your paper is free from any fabricated data, means cooked data. Some people, they make up the data, they write the paper, and end of the day, the paper may be retracted once it is published. So be very careful and make sure that your paper is free from any falsified data, any manipulated data, and make sure that your paper is free from any plagiarism. Because once you submit your paper to elsewhere, the elsewhere will automatically subject your paper to authenticate, where they will be able to find out the sentence similarity and plagiarism. If it goes beyond a certain percentage, the paper will be returned back to you. Most of the time, because of high competition, the paper will be desk rejected. So you have to be very careful that the paper is free from plagiarism. Otherwise, even your paper is accepted later on, your paper will likely be retracted. That also you have to be very careful. And also you should never practice salami slicing, breaking the paper, a single paper into 4 or 5 least unit publishable. Sometime, the reviewer or editor might comment that this is the follow study of the author. And the author should have published this study also along with that study. We do not understand why the author wants to submit a separate, a small publishable unit article. So it is a salami article, hence it must be rejected. So such kind of comments you may receive. So you should avoid employing salami slicing in your study. And also you should never, yesterday I told you, cite any article from predatory journal, any article from hijacked cloned journal. For example, your paper has 50 references. Among 50 references, around 10, 15 references are from predatory journals, International Journal of Recent Advances, so, like that. There's so many predatory journals are there in India. Number of predatory journals. I'm not saying that don't refer it. I'm saying that don't see it. Don't have a look at it. Throw it. Don't cite any references from predatory journals. Otherwise, there is a machine learning system from elsewhere that is known as MARS, Machine Learning Referencing System. They will subject your paper to machine learning referencing system, and they will see how many references are authentic, I mean valid, and how many references are invalid. For example, you are submitting a paper with 50 references, 44 out of 50 references are invalid. Means all are from predatory sources, non-indexed sources. 200% your article will be desk rejected. And also don't cite any paper from cloned or hijacked journal. Sir, how we can find out cloned journal? That experiences over the years you will come to know. Once your paper is published in a cloned journal, it will be never indexed to be PubMed Medline. It won't appear in PubMed Medline. It won't appear in Web of Science. Yesterday I have shown you, and it won't appear in Scopus database. It means something wrong. The journal is there in databases, but my paper is not. Try to go for a credible journal. Don't avoid publishing your paper in a predatory journal. And in your paper, there should not be any guest author, a gift author, or ghost author. So guest author means they have not done anything that you are including them in the paper. Gift author means without their knowledge, you are gifting. Suddenly you are including their name in the paper and you are making them surprise. That is also misconduct. Ghost author means you have done everything, but your name is end of the day is not available in the paper and someone is publishing. For example, you pay 10,000 rupees to some company, they do all the project for you and all the paper they write for you, but end of the day, their name will not be there, your name will be there. So they are the ghost author. So these practices, you must avoid it. Don't give, if you are publishing a paper, just restrict to number of authors, your name and your guide name. That's all. If you want to put a third person name, that person must have done a significant contribution to your study. Unless otherwise, you don't include any person name in your paper, that will dilute the quality of your paper. Suppose if you're applying for a postdoc fellowship, if you're applying for a PhD program to foreign universities, so such kind of paper having only one or two authors, that always has a very valid justification, and you cannot include 5-6 authors and you may be the fifth author or sixth author. It means your contribution is very less. So try to become a key author. So there are different types of publication. Dr. Jagdish sir must have discussed about this one. Original article, short communication, rapid communication, brief, ultra rapid communication, review article, state of the art review, letter to the editor, commentary, clinical case report, methods, technological or laboratory notes, book review, so on. So among these, we are going to discuss about today the research and review article, original research or review article. So coming to the point directly, how to avoid desk rejection of your manuscript. So most of the informed students, they might not be aware of desk rejection. Desk rejection means you have done your research. Good. You have written your manuscript. Well, and you have submitted it to your target journal. And almost immediately you get a reply of no. It means your article has been desk rejected. You have been desk rejected. For example, today evening, 7 o'clock, you are submitting a paper for consideration by a indexed journal. And same day or next day, I mean tomorrow, you get a reply, decision on your manuscript. We are sorry to inform you that your manuscript has been rejected from publication by our journal. There are several factors to reach this decision. Don't be disappointed. You can find a suitable another journal for submitting your paper. Wish you all the best. So these are the common email that people are receiving. Why the manuscript is desk rejected? What are the reason? Editor, because of lack of time, they don't write anything. Just they see your manuscript, see the language, see the uniqueness, and see the methodology what you have performed, see the result, and what conclusion you have made based on the results what you have obtained. So based on all these factors, they consider your manuscript either to subject it for peer review or desk rejection. So if you get a desk rejection, psychologically, it creates a problem, because after a long time you're writing a manuscript and finally it is rejected by a journal means there are a lot of reasons. Maybe it may not be in the scope of the journal also. It is your mistake. So this is what I'm going to discuss now. A desk rejection means you did not make it to peer review, did not get past the first screening. Hence, it is bad to go for you and your team. Of course, certainly it stings because you are not alone. You are with your team. So definitely it creates a psychological problem. So how to avoid? What are the potential ways to avoid desk rejection of your manuscript. Number one, edit or at least proofread your English. This is most important thing. As editor, our first look into your paper is your abstract. After having read the title of your paper, we will directly enter reading your abstract, and we will see whether your abstract is structured properly or not. Abstract should have four components. The first component should be background. The second component should be method. The third component should be results. And the fourth component should be conclusion. There won't be any discussion and there won't be any references in the abstract. So in the editor will expect you to write the problem statement. The problem statement. So you have to clearly pitch out the problem statement, and based on that one, you have to write the research gap. The research gap, it is still remain unknown. It is not clear. So you have to write the gap. Therefore, the present study has been designed to investigate. So the background itself should have a research problem, research gap, and aims. No need objective, aims. The present study has been designed to evaluate the anti-diabetic effect of newly synthesized compound. So in the background, you can include one or two sentences. Because of the restriction of number of words in the abstract, it is important for you to consider number of words as well while writing the abstract. So in the background, you write the problem statement. Diabetes mellitus is the leading cause of mortality worldwide, and its prevalence is incessantly increasing. The existing medications are unable to manage the disease completely, or unable to cure the disorder. Hence, the present study has been designed to synthesize and develop a new compound like that. So you have to give a background with the problem statement and research gap along with the aims. Then method. Method is the second largest portion in the abstract, and you should include a newly developed method. I mean the novel method or important method what you have performed in your study. Then result. Results should always show some numerical value. For example, p-value, percentage changes. So some kind of numerical value should be shown in the result part of the abstract. Then conclusion. Conclusion should exclusively based on the results of your study, not the assumption of your study. Just you can it may be it not like that. It is because of this one. So you write the conclusion based on the results, and also even if some surprising findings are there, that also can be included in the conclusion. So totally it should not exceed 250 words. And some journals like British Journal of Pharmacology, they may ask you to come up with 150 worded abstract. It means more focused one. Some journal they might need to come up with a structured abstract. Some journals they might need you to come up with a unstructured abstract. So these all things are very, very important, and you should as an editor we proofread your English. I mean we go through your abstract. Suppose if something is wrong, it is not clearly written, it is very poorly written with a lot of grammatical and typographical error, right away your paper will be desk rejected. The editors do not have time to improve your paper, the language. It is not a duty of the editor. So you have to at least proofread your English, and you have to read the article minimum 50 times. Minimum 50 times before you submit your article to consideration for any journals, by any journals. Don't submit immediately that, okay, now no more I can read. No, you have to have a patient. Take a printout of your article, sit on the table, very comfortably take a cup of coffee or tea and read the article and keep on reading. And once you are reaching to the stage where, oh, there is no grammatical mistake, it is absolutely correct. Whatever the points I thought in my mind, everything are included. Even the limitations of the studies are included, hypotheses are included, and null hypothesis, the rejection of null hypothesis is very clearly mentioned in the discussion. There are differences between the group, and very properly we applied the statistics, and the statistical analysis and results are very well expressed in all the figures, and all the figures are given with the statistical comparison. So this, now I'm okay. Now this manuscript can be submitted. So you have to reach to this level before you submit your manuscript. Second, you use active voice rather than passive voice in the manuscript. Propranolol is a beta blocker. That's all. It has been reported that propranolol is a beta blocker. No, it's not necessary. Directly, you write a direct sentence, a simple sentence in active voice rather than passive voice. I eat pineapple. That's all. Pineapple is eaten by me. It is not necessary to write in passive voice. And have all co-authors check it and sign it off. And for example, all co-author means, in my opinion, you and your guide only. If it is a PG work, if it is a PhD work, definitely you and your guide. If some co-guide is there, yes, the third person should be there. Don't include fifth, sixth person, seventh person. That person did for me histopathological study. That person did for me statistical analysis. That person improved my manuscript grammatically. So I have to include all of them. I have to please them. No need to please anyone. As long as the research is concerned, the person who has done the work, the person who is guiding the student, only two names, that's all. If it is a multidisciplinary work, if it is a multi-center work, if it is a collaborative study, yes, you can. But during the MPharm and PhD, these collaborative studies are not possible. They can be done only after you establish yourself in the field. Make sure that the manuscript has a relevant title. The title may be descriptive or declarative or interrogative. Descriptive means you are not declaring the result. Paracetamol, effect of paracetamol on fever. Declarative means you are declaring the result in the title itself. Paracetamol relieves fever. Interrogative means there is a question mark. Does paracetamol relieve fever? There is a question mark. Most of the time, the research article won't carry the question mark. It may come up with, you may come up with a research article with a descriptive or declarative title. Still, many journals might expect you to come up with a declarative title. Title itself, you declare the result. So make sure that you what kind of study, whether it is a cross-sectional study or longitudinal study, or it is a cohort study or pilot study, whatever it may be, just mention in the title itself. Or experimental study, or you are using rats or mice or guinea pig or rabbits, whatever it may be, everything should be there in the title. Make sure that you carefully review the guidelines, since certain publication may require the study design to be included in the title. This is what a study design means: a cross-sectional study, a longitudinal study, a multi-center study. So like that, kind of the design also should be included in the title. So yesterday I told you, in the title should be, you should be very specific. Rather than saying salad, say cucumber. Specific. That is most important thing. Make sure that the abstract is concise, but it contains the essential information. So I told you, you should make sure that the abstract explains enough detail and how the study includes the new knowledge to the field. So you can say, to the best of our knowledge, if it is a new knowledge, to the best of our knowledge, this is the first study demonstrating. It's excellent, but it should be really a first study to demonstrate. It is not like that simply you can include. And you should put the abstract in the right structure. I told you structured abstract, non-structured abstract. Structured abstract will have background, methods, results, and conclusion. Non-structured abstract will have all these components without any subheadings. That's it. And you should always come up with a graphical abstract. Graphical abstract is an abstract which is drawn, not written, like a simple abstract. So you draw the abstract and make the readers understood of your study and its outcomes. So a very good graphical abstract will find a very confident place for you to get your paper considered. So furthermore, look for a graphical abstract which can provide a contemporary visual touch that appeals to those with a shorter attention span and therefore those who are more visually oriented. Some people, they are reluctant to read anything. They just want to see and observe the content. So for these people and for everyone, if you are able to come up with a graphical abstract, it is very good. Some journal, they give an optional for graphical abstract. Don't take it optional. Take it a key component of your manuscript. Make sure that your study should contain a novel or innovative aspect. Something, at least 5% novelty should be there in your study. Then only your study can find a place for acceptance. Otherwise, it is desk rejected. So it is outdated study. Already a number of studies have been reported, whatever is being reported in this paper. So this paper is not providing a new knowledge to the field. Hence, it is desk rejected. Because the editors, of course, want to publish new things in their journal. Then only they get more citations. If they get more citations from your article, their H index will go. Their impact factor will go up. For example, pharmacological research, as far as my knowledge goes, in 2000, its impact factor was 1.7. Then progressed into 2.5. Then 3.2, then 4, then 5, then 6, then 8. Now our journal has an impact factor 9.1. Still we want to make it to 10, then 15, then 20. So how this thing can happen? Can happen through only by publishing a novel work, unique work. Okay, so you should never cite any references in the abstract. If you are citing any references in the abstract, right away it will be desk rejected, because citing references should not be done in the abstract. In the introduction, you should use recent citations. For example, don't write introduction for more than 500 words. Just a crispy and focusedly written introduction. The introduction, suppose you are referring 15 references, for example, you are citing, out of 15, at least 10 references should be from recent sources. And the IMRAD style, introduction, materials, results, and discussion. So you have to follow according to the journal guidelines. Make life easier for the journal editor. You can be desk rejected if the paper is unusual or do not adhere to the journal requirement. Peer review requires time and effort. For example, you are submitting a paper and editor, it is convinced by the editor. I mean, you have convinced the editor. Everything is good, well written, and the novel concepts are there, advanced methodology is there, references are high quality references, all are from PubMed Medline, excellent, outstanding, and the paper is very well structured. So ultimately, your paper will be sent for peer review. And getting a peer review is not an easy task. As editor, we struggle to get two peer reviewers. And for that, first we will send your paper to 10 peer reviewer. Out of 10, they will decline it because they do not have time. It is a voluntary service. So they are not supposed to, accept the invitation. Then again 10 reviewers, again 10 reviewers. Recently, one journal, it is an open access open journal, not a single blind or double blind, open journal. And their journal, I submitted the paper. They sent my paper to 251 reviewers. Out of 251 reviewers, only one reviewer has agreed to evaluate the manuscript. And that reviewer has accepted the manuscript, recommended it for publication. And the second reviewer, the journal did not get it. And so still it is pending. It has been around 4 or 5 months. So getting a reviewer is very, very, very tough task because it is a voluntary service. So the editor wants to send only those papers which can pass the gatekeepers. If the editor has a doubt on your paper that it will unlikely be processed successfully, then definitely your paper will be desk rejected. So do not communicate the editor frequently. For example, you have submitted your paper 3 months, you do not get any response. You keep quiet. 4 months, you keep quiet. Then 5th month, you can send a reminder to the editor that I have submitted my paper in the month of August. So far, I have not received any response from you. Can you please expedite the process of peer review so that our work that shows some new knowledge to the field will be published in time, because publishing article in time is of utmost importance in science. So you can write politely to the editor. The editor will respond, oh, we are very sorry that we will send a reminder to the reviewer. So because they are handling too many manuscript, your manuscript is one among them. Your hypothesis is made based on the research gap. So your hypothesis must be very clear. You must have a strong research motive. The conclusion must make a sense. And your conclusion should be written based on your own result, not based on your hypothesis. The study should use acceptable outdated. For example, you are writing DNA is a double-stranded helical molecule. So bracket, you should write Watson Crick 1953 only. You cannot write any other name, because some people might have written that one. You cannot write Balakumar 2024. It is not good. So you have to write a pertinent, you have to cite a pertinent references. For example, G protein coupled receptors play a key role, and around 50% of the cardiovascular drugs are acting through G protein coupled receptor. Cite Gilman et al. The Gilman who has got a Nobel Prize for the discovery of G protein. That shows your experiences. Nitric oxide is a powerful vasodilator. It is also known as endothelium derived relaxing factor. It is released from tunica intima, the inner wall of the blood vessel, that causes vasodilation of the blood vessels and maintain the vascular tone. Write the reference of Furchgart and Sawatsky. Murad and Ignoro, for that they got a Nobel Prize in 1998. You cannot write simply any local references, because any statement can be written in any paper. So the accuracy of the references, what you are citing in your manuscript, it all depends upon your expertise. So you should keep on reading the quality article. You should keep on referring the quality journal. Don't refer any underestimated or predatory cloned journals. Figures and captions should be very clear. The figure should be standalone. For example, a figure is there. As soon as I see your figure, and it should be standalone. Very clearly, there should be the caption. This figure shows this one, and there are differences. P-value less than 0.05 versus this comparison with this group to this group. So like that, the figure caption should be very clear, and it is very much necessary. Don't include any figure in the manuscript. Upload them separately. Upload them separately. References should be very comprehensive and in the right format. If the journal asked Vancouver, so use Vancouver. American Psychological Association, American Psychological Association. American Medical Association, American Medical. If it is Harvard, Harvard style. So just to check the journals and see the recent volume they have published and see the references. Nowadays, your paper, your way is there. You can submit your paper in any format. But once you are submitting a revised manuscript, you have to adhere to the guidelines of the journal, and not too few and too many references. For example, you are writing a research paper and having only 10 references. What does it mean? And you are writing a research paper, you are having 140 references. What does it mean? Don't go less than 30, don't go more than 50. So keep it from 30 to 50, not too few references, not too many references. And a good cover letter can help sell your study to the journal editor. A one-page cover letter, Dr. Jagdish must have discussed this one. So you should state that the paper is original and unpublished, not submitted to any other journal. All authors have agreed, and outline of the work and conflict of interest, any financial conflict of interest, and the source of funding. Even if your work is not has not received any funding, you write down, and all the authors, that is credit author statement, that is contributed roles taxonomy, credit author statement, the first author, conceptualization, methodology, done the experiment, written the first draft, approved the first draft. So second author, the corresponding author, supervision. So all things, whatever the contribution is there. So a very clear one-page cover letter should be there. If your manuscript is rejected, treat the rejection as your fault, not the fault of the editor, because most of the editor, they spend time on your manuscript. If they think that your manuscript unlikely will be processed, will be rejected, or if any of the reviewer has given a negative comment, because there will be a confidential comment to be sent to the editor by the reviewer. Maybe the comment that is sent to you may be good. The paper is interesting, like that, something they might have written. But in the confidential comment, they might have written anything, something adversely on your manuscript. So based on that one, the manuscript may have been rejected. So these are the submission checklist. Make your cover letter ready. Your graphical abstract should be separately ready. Abstract, a separate page. So abstract should be standalone. Then manuscript, start with the title, then author detail, affiliation, then corresponding author, then again abstract, then below to the abstract, there are 6 keywords. Make sure that all the keywords are retrieved from PubMed mesh database, medical subject heading. From in that database, you should try to retrieve the keywords. Then after that, you should include the list of abbreviations. Suppose your manuscript has more than 10 abbreviation, like PPAR, gamma, peroxisome proliferator activated receptor gamma, DNA, like that 10 abbreviations are there, then it qualifies for a separate list of abbreviation. Yes, you can provide. Then you should include a separate page for highlight. Highlight means a 5 bullet point. Each bullet point should have a 80 characters with space, and you say, well, what you have done, what was the result, and what is the conclusion. That's simply a 5 bullet point. If anyone want to see your highlight, and they should be able to understand the entire work within 1 minute. Within 1 minute. Then start with the IMRAD, introduction, materials and method, including protocol, results, discussion, conclusion. Conclusion is the part of discussion. Once you end up with a conclusion, then provide acknowledgement, in which any funding source, anything acknowledge you want to acknowledge. Don't acknowledge simply that we are thankful to this person for his support, for his help. So it is not necessary. If anything that the work is done by someone else, or someone has given some ideas, so you acknowledge that person. Then conflict of interest statement. Most of the time, the PG student or academic work, they do not have any conflict. And the credit author statement, I told each and every author what they have contributed, so that thing should be there. And the credit means contributed roles taxonomy. Then funding detail. If no funding is there, then you should just mention it. It is very much important. Our internal funding, if you get it, then just we are thankful for the internal seed grant, internal seed money. Like that, you can mention it. And then references. I told you all the references that you are going to cite in your manuscript, they should be from PubMed Medline. Don't cite any references other than PubMed Medline, because you are from a biomedical field. And most of the articles should come from PubMed Medline, or at least if suppose 1 or 2 from coming from Web of Science, but that is not available in PubMed Medline, does not matter. It's okay. But don't cite any cloned, hijacked, any predatory third-class journal. So if you cite that kind of papers, if that paper is invalid, and your paper also is considered invalid. So once you given all the references, give a legends for, captions for figure and table. Figure 1, figure 2, figure 3, figure 4. Don't include any figure. Upload them separately. The 5th point, you upload figure 1, figure 2, figure 3, figure 4, figure 5, figure 6, table 1, table 2, table 3, everything upload them separately. If there is a need for you to include a box, Means, for example, 1.5 billion people have been affected by 1 billion people have been affected by hypertension. In 2040, it may be increased to 1.5 billion people. So such kind of a key point that you want to provide to the audience, yes, you can include the box that is separately. When we publish a paper along with Dr. Jagdish for pharmacological research, prevalence and prevention of cardiovascular disease, we have given around 10 boxes, and all the boxes were published inside the manuscript, and all are very highlight points. And that paper I told yesterday got cited more than 800 times. In another 1 year or 1.5 years, it will reach 1,000 cited paper. I mean, a highly cited paper. Definitely that paper will reach 1,000 citations because of its high reachability and readability by scientists throughout the globe. Then author declaration form. Yes, you should declare all authors have read and approved the manuscript, and we agree with the order of the author and title of the paper, and we don't have any conflict of interest. So separately, conflict of interest form should be signed and uploaded all the author, including their ORCAD. That can be automatically, when you are uploading the manuscript, it will be automatically uptaken. They give optional for ORCAD. Don't take it optional. Provide the ORCAD. Nowadays, ORCAD, if you have the paper, that can be directly linked to Scopus. And if you have ORCAD linked paper, that can be directly linked to Web of Science. So directly you can export the paper from ORCAD to Web of Science. So don't ignore that one. Open ORCAD today itself, open researcher contributor ID and enter all the detail. And whenever you publish a paper, you just upload it. And sometime, automatically it will ask, the system will ask, one paper is knocking the door. Can we accept it? Please approve it. Just approve it. And it will be entered into your account. Later on, if you want to export to the Web of Science, yes, you can easily export all the paper from ORCAD into Web of Science. So, dear audience, coming to the last point, it takes commitment, persistent to prepare a strong article. You have to sit minimum 4 hours on the table. It is not like that 10 minutes you are sitting and you are attending the mobile phone, you are attending WhatsApp and responding people, good morning, I'm very good. I have also taken lunch. You have also taken lunch. No need. 4 hours. Throw the mobile phone, sit on the table on the computer in front, and you start writing the article. It takes commitment and perseverance. Daily 4 hours and 1 week you can write article. Within a 1 week you can write article. So it takes a commitment and a persistence to prepare a strong article that will be accepted for review and ultimately for publication. When I receive a paper, when it is free from any error, any grammatical, any typographical error, and it shows the novel concept, the conclusion is very well written, the references sections are ultimately structured up to the international standard, and all the diagram table are outstanding, there is no way. We have to send the paper for peer review. There is no way. Why the paper has to be rejected, just rejected? Even when you believe that you have done everything correctly, remember that the journal editors are individual. They have choices and preferences. And sometimes you may catch them on a bad day. So it all depends. But don't worry. A good paper will find a way. If your work is strong, look for a different target journal. Be open to criticism and try to receive the comments from the reviewer. If you agree with the comment, you concur with them. If you do not, you politely tell them that we are unable to accept the comment of the reviewer as reported previously. Show the evidence. Show the evidence very clearly. Make the necessary revision and try again. Writing a good manuscript is not an easy process. It takes commitment and perseverance. You need to sit for 4 to 6 hours uninterruptedly and non-stoppingly. Your paper is worthless if it is not cited. Even see the outcome of the publication is research is publication. The outcome of the publication is citation, I told you. And the outcome of the citation is H-index. Nowadays, suppose if you want to have a job in Saudi Arabia for a professor position, they will ask you what is your H-index. If you want to enter in Malaysia, some good university, they will ask you, what is your H-index? So H index is calculated based on two parameter, number of paper and number of citation. For example, if your H index is 20, it means minimum you have published 20 papers and all the 20 papers have been cited minimum 20 times and maybe more than that also. So if your H index is 15, that's all. You are eligible to become a professor in Gulf countries. So 15 reaching a H index 15, that I'm telling 15 is a Web of Science H index, not a Google Scholar H index. So Web of Science H. Nowadays, the NAP team, they divide the citation, Web of Science citation plus corpus citation divided by 2. Web of Science H index plus corpus H index divided by 2. So that formula is being followed nowadays. So these are the gatekeepers, Jagdish sir must have told. Journal administrative staff, receiving editor, editor-in-chief, associate editor, and reviewers. So coming to the conclusion, you should select the right journal, review the submission guidelines, prepare a high-quality manuscript. If it is coming for revision, address the reviewer comments very politely, not arrogantly, and if you accept it, excellent. If you do not agree with them, respond to them with the rebuttal very politely. Understand the subscription mode. See, some journal, they ask you to pay money, open access. So go to Serpa Romeo, where the open access policy will be there. Some open access may be green open access. Some open access may be gold open access. So green open access, you do not need to pay money. The society will pay money. Gold open access, you have to pay money. Platinum open access, definitely you have to pay money. So by using Serpa Romeo, the platform, you will find out what kind of open access the journal has. So accordingly, you can move forward, but don't try to spend money for publishing your paper. I have published around 140 papers. So far, I have not spent even a single rupee from my pocket for publishing my paper. And also, I will not spend money throughout my career. So no, it is not necessary to spend money. You can submit your paper through subscription mode, where it is very tough to get your paper go through, but still no need to pay any money. Check the journal indexing, whether it is indexed in Web of Science. Go to Clarivate Journal Master List, write the journal, and see whether the journal is Science Citation Index expanded or not. If so, it is Q1 or Q2 or Q3 or Q4. Even some journals, Emerging Sources Citation Index, it is not, their journal also might have impact factor, not might have, their journal also have impact factor, and maybe in Q1 also. So make sure that the journal is from Science Citation Index expanded. Build a strong authorship team. See, we used to publish Dr. Jagdish, myself, Dr. Fred Senator, the morning the session you had, we had combinedly published a paper in 2017-18, and also we edited a book. Dr. Senator is a very dedicated person like Dr. Jagdish, and they are working for US FDA. They are meticulous people. Working with them is not a joke. It is a very, very tough task. And you need to spend a lot of money, and they ask queries like anything. You need to address their queries. You need to convince them. It is not an easy task. Present original and significant research. Be persistent and patient. The peer review process can take time, and you should contribute constantly to the science. Sir, I become the professor. I already published 40 papers. Why should I publish now? Leave it, nothing is there. I already become the professor. Why should I? No, you should constantly contribute to the science. See, the age of Jagdish sir, very, very senior person, still he is contributing. And we have already planned yesterday. Sir, we need to move for a special issue for Journal of American College of Cardiology that has a 20 impact factor. So we need to work on it along with Dr. Fred. Yeah, we should do it. That one definitely we should get it done. So even at the level of having 50 years experience, still he is very thirsty to write a paper. What about you? You are maybe 35, 40 or 45 or 50 years old. So you should contribute constantly to science. So what the editor might look into your paper. The first, does the manuscript have the attractive title? Is the work is having a uniqueness? Is the paper is from a reputed lab? Will it receive attention for more readers? Then only the more chances for getting citation, and for them the impact factor will go up. Does the manuscript strictly follow the guidelines of the journal or did not follow? Is the submitted manuscript well written? So everything the editor will see. If your manuscript looks messy, it might sometimes be presumed that everything must be messy. So avoid publishing in a predatory and non-indexed journal. Enhance the impact of our research by publishing in SCIE SSCA is not applicable for us, SCAE, and enhance the citation and H index of you and the institution where you are working. So your paper's likelihood of acceptance increases if it describes the field advancing the research, if it is expertly written and formatted, if it speaks in a clear and concise manner, if it follows ethics and free from any scientific misconduct. I told you FFP, plagiarism, FFP, falsification, fabrication, plagiarism. So it should be free from FFP. Otherwise, see, so 80% of the manuscripts are retracted because of, and this figure is stolen from previous sources. And this figure is used by the author that was already published by another author. And there are 20 more than 20 tables they have copied from these sources. So these are the reasons for the retraction. See, retract one, if your paper is going to be retracted, they will not retract within a day. They will send a letter to you. And they will send a letter with the evidence. Not a journal. The publisher will send it. Not the journal person, not editor. The publisher will send it. Dear doctor, there are some flaws in your manuscript. Figure 3, figure 7, figure 8 that you have copied from the following sources. It is not good. So you please justify. We are going to retract your paper. And some author, they respond, yeah, it is our mistake. Very sorry. Okay, the authors have agreed for retraction. Some author will say, they will not say anything, just keep quiet. Still, they will retract. They will say that the authors did not respond. So, when you get a letter from the publisher, very painful one, and it is not only good for you, for the institution also, because along with your name, institution also affiliated. So these all things create a problem. So please try to publish any paper that is free from scientific misconduct. Initially, you may struggle for publishing your paper. When you keep on trying, definitely you can get through, and you can get a successful publication of your work in a journal of international repute with the globally indexed databases. Thank you very much, the organizers, Professor Ramesh and Professor Rajasekhar, for their kindness inviting me to deliver the presentation on this particular topic to the audience of high interest. Thank you very much. Have a great day.
Thank you so much, sir, for sharing your wonderful experiences and thoughts. And I think this is a really wonderful session. I believe, although I missed it because there is an inauguration happening for Vishnu Cup. I think the session is open for questions. Any one of you, please, you can shoot up your questions and check in online also.
Dr. Balkumar, it was so exciting, all this thing, the presentation. I have a question regarding retraction. I think many of you people may be interested to know that why the retraction is done so lately, saying that citing some figures, tables that have been copied either from other sources or from their own publications, why there is a late realization by the publisher, but not before. When they submit it, of course, peer reviewers would have pointed out, but may not be possible immediately. But there should be some mechanism where they should be able to point out such image copying or copying plagiarism or falsification, why it is not being discovered before goes into press. Any idea?
Yeah. Your question is very valid, sir. This is what we also wish for, because when the manuscript is processed by the peer reviewer, that stage itself it can be caught. But you know, the machine learning and the deep learning services available currently, the artificial intelligence, they are unable to find out whether the figure is copied from other sources or not, because all the figures that the people submit, it is a protected one. And once you publish the paper, once it is available public, and the people are referring the paper, and they are coming to know why they have used my figure, then the concerned author send a complaint to the publisher under the journal, and accordingly the action is taken. So currently we do not have any system to find out the plagiarized figures, copied figure. So that is the lacuna, and that is the need of ours, sir. There is no way for us. Only after the paper is published, because of the malpractice in using the duplicate figures, and once we come to know with evidence by the concerned authors, after they file the complaint, then only we are able to take it forward to the publisher, and the publisher verify it, and accordingly it is communicated to the authors for concerned action.
How about artificial intelligence detection? Yesterday we were talking about artificial intelligence, a role in review of literature. Is there any such artificial intelligence detection of such image fabrication?
Yeah, image fabrication is not available, sir. Most of the histopathology, even Western blotting, most of the time, they fabricate on Western blotting and this histopathology. So that image manipulation is unable to detect it out. Later on only it is possible. All the plagiarism and everything they are able to find out now through Turnitin and Authenticate. But the image manipulation, image duplication, that is a great problem, sir, now.
Good morning, sir. Sir, I have a question. You told that most of the citations should be from PubMed. So, but PubMed journals don't have open access. Like I have tried for many number of journals to search, but PubMed is not having open access. Can you suggest any other ways other than subscription to get access? Other than subscriptions for PubMed.
You are correct. You can go to PubMed Central, where you will get a free full text articles. Second, in the PubMed, whatever the article comes, the abstract is freely available. So through abstract, you will come to know what is the methodology they used and what results they got, and what is the conclusion. Based on that, you can easily cite a paper. That is more than sufficient. Suppose if you want to explore the methodology, what they have done, then you need a full paper. For that, you need a reprint. For citing a paper, abstract is more than sufficient.
And sir, how can we identify that whether one particular article is retracted or it is having a hijacked score or something like that?
Yeah, it is automatically written top on it. Let me demonstrate it. For example, are you able to see the Chrome?
Sir. Yes, sir.
Yeah. So now let me open PubMed. Automatically, see, I just put retracted. In the title itself, it is shown. See, retraction note, compassionate use of quantum magnetic resonance. Retraction note, ethanol extract of this one ameliorate cough variant asthma through this pathway. So it is a Chinese article. So now I'm opening and see the retraction of this article, retraction in. So just if you open this article, see the abstract is there. And if you open the full text through Springer link, automatically, it will be directing. So this is the article which is already retracted. See here, it is written retracted article. So suppose when you are referring, the article might not have been retracted. When you are submitting the manuscript after 2 or 3 months, it might have been retracted. So you have to check all the references again, whether it is retracted or not. So you have to check it. So that is most important thing. Now see here, why they have retracted. The editor-in-chief has retracted this article at the request of the author. Upon further analysis, the author discovered that due to an oversight, protein band images from animal experiments were mistakenly included in the cell experiment protein band image result of this article. This has led to the key data error and deficiency in interpretation, which may affect the accuracy and reliability of the research finding. In this case, the author themselves, they send a note to the publisher that please retract it. We have mistakenly used images for interpretation. So the reason also will be available for a retraction note. Not only this article, there are so many articles that you can find out through PubMed. So once you have included any references in the article, that time it might not have been retracted. But once after completing your article after 3 or 4 months, again you have to check all the references. Is there any retraction or not? But once you submitted the paper, once it is published, later on, if any of the references are retracted, does not matter. It is not your mistake. But nowadays, around 6 to 10% of the submitted manuscript have retracted paper, the people say here. Now see here, prevalence of testosterone, something they retracted. So this is the paper they retracted. So everywhere else you can find out. In the title itself, there is a note, a retraction notice. It is again from author from Madurai Kamraj. So this one, the article is retracted. Control minus. So here they clearly mention this. There is a retraction notice for the article. And if you go through the article and you can find out the retraction, what is the reason for the see here retraction notice. Retraction notice. So here there is a authorship changes were made after acceptance of this paper. See the reason. Authorship changes were made after acceptance of this paper. An author was removed and Arivalagan Pugalenthi was ordered as a co-author without validation and authorization. There was a significant increase of citation of the paper authored by Pugalenthi between the original submission and the revised version. So now here what they did, there is an author misconduct. Initially, they provided a set of author, then they removed one author and ordered another author as a co-author. So this one, in addition, acceptance of the article was solely based on the positive advice of the reviewer who were closely linked to this one. So they put this person name and the reviewer knows that person and the paper was accepted. So everything they publish it, the reason for retraction. It is not only for figure manipulation, this author manipulation also. In this paper, there are authors and they removed one author and put another author. That author is known to the reviewer, for example, and the reviewer has given a positive comment and the paper is accepted. So this compromised editorial process and breached the general policy. The authors disagree. So they sent this retraction note to the author, but they did not agree. No, we don't agree that one. So you can see the reason also for retraction, and it is not only image manipulation or plagiarism. Once you submit the manuscript, don't delete the author name, don't accept new author name. It is very, very difficult. Sometimes, we have 5 authors and one author suddenly withdraw the paper. Sir, I do not want to become the author of this paper. There are problems. So it is a problem. It is really a problem. So that's why I'm telling, don't go for multiple author. You and your guide, that's all, enough. No need to go for a 3rd or 4th or 5th author. So like that, so we can show a number of examples, dear student.
Thank you, sir. Thank you so much.
Yeah, you are welcome.
Good morning, sir. I'm
Good
Sheikh
morning.
Naeem from Polarity Institute of Pharmacy, sir.
Yeah.
Sir, I have a question like in the discussion, what are the precise points should be written in the discussion part, sir?
Okay. So you should start a discussion with your findings, major findings. So in the present study, we have performed this one and we have observed this one. That's all. The first paragraph started. Then why you have observed? What is the reason? So you just include the existing literature, whether it is in contrast or in association. So everything, give a critical discussion of your methodology and result. And don't try to forget including limitation of your study. Every study must have a limitation. If you do not point out the limitation, the reviewer will point out the limitation. So you just include the limitation of the study. Then finally, conclusion. In the conclusion, you have to include 3 points, minimum 3 points. In the present study, we observed this one. Based on based on these observation, we suggest or we reveal or we propose what we propose that one. However, nevertheless, further studies are needed because every study should open up with a new study, open up for a new study. So that should be included. So that's all about discussion. Separately, I think we need to is a separate session that how to write a discussion. Maybe in next time we will try to discuss how to write a discussion separately.
Thank you, sir.
You are welcome. In the discussion, again write in the present tense and the present perfect tense. Don't write in past tense, and also cite relevant references and should be the healthy discussion, starting with the finding of your study and what all these findings mean. Then you just discuss one by one. Then whether your results are in accordance with the existing report or not. If it is in contrast, you have to give explanation. Then give a limitation. Then don't go for any sweeping statement. That should not be the part of discussion. Then conclusion. Then the conclusion last line should be, I told you, nevertheless, further studies are needed. Then you have to include one more line. Although further studies are needed, the present study provide new knowledge and advances the current knowledge. That point is very much important. If you say that further studies are needed, the reviewer will ask you that go do the further study, resubmit it. Although further studies are needed, the present study contributes to the better understanding of the current knowledge. So that point should be there at the last point. Don't forget it.
Once again, sir, we extend heartfelt thanks to you for your time and sharing your all expertise, knowledge with us. It always a great honor to listen you and learn from you always, and looking forward to you in upcoming sessions too. Learn from you.
Sure,
Thank you very
sir.
much. Have a great day, sir. Thank you very much.
My pleasure, and thank you very much. Once
Yeah.
again, I thank the organizer for the opportunity to interact with your
Yes.
well-established team. Thank you very much, and
Yeah.
I look forward to meeting you all in person next year.
Definitely. That's sure. Definitely for sure, sir. Yeah. So students, now we'll break for 10 minutes. The tea is arranged in canteen for you. You can, sir? Yeah. And actually, madam is waiting for you. Right, please. Actually, she joined now. So I requested her time for 10 minutes so that we can have a quick break, go and come back sharp, right? And another session is very important. Dr. Subhada Nagarkar, madam from Pune, she is going to talk to you on and enrich your knowledge how to identify the predatory journals, which is most important, and what is the research ethics, right? Please come back within 10 minutes, right?